Welcome to Molecular Genetics Part 1. By now you've watched a DVD on cracking the genetic code, and in that DVD you've become somewhat aware of the characters on the screen here and the process of identifying the genes in the human genome, that race that basically was fought between private enterprise and the public purse. Directing that public purse was Francis Collins, and of course the initial structure of DNA that you're familiar with, the double helix. The Nobel Prize was won by Francis Crick and James Watson, these two gentlemen at the top at Cambridge, playing with their molecular structure. I find it interesting that since Watson and Crick received that prize, there's been a lot of evidence to suggest that this young lady, Frances, why is everybody Francis today? This is Rosalind Franklin. I'm going to write Franklin down here. Rosalind Franklin. You may have heard of the Franklin Institute. She was a lab assistant in the laboratory where Watson and Crick were actually researching DNA structure as a nucleic acid, and she was doing X-ray crystallography, looking at strange pictures of the molecule itself, and she, perhaps more than they, deserved to get credit for identifying how the bases in DNA would have to fit together in order to come up with the double helix structure. The sad part of this tale is that Miss Franklin happened to be a female working in a science laboratory in England, and I shouldn't pick on England here because this was true pretty much of the world at that time, that women were not given an awful lot of credit for their findings in science. She also happened to be Jewish and young. All of these strikes against her, and the saddest part of it, of course, is that she ended up dying young of cancer, and during her life never did really get recognized for the role she played in it. Consider this page somewhat of a an attempt to somehow whet your appetite to go beyond what you find in simply a biology textbook on molecular genetics. You can get the absolute basics out of that textbook, but what you will probably miss is the fact that there's been some amazing people and some amazing stories involved in this whole field of molecular genetics. It's probably the cutting edge field in biology today. So much being done in this area, and it's really far better to, try to go beyond simply what's in that textbook. So consider reading about Rosalind Franklin, or you might consider reading The Language of God by Francis Collins. He was the chap that led the American team in that Human Genome Project. I find him interesting not only because he was a great physician and I shouldn't pretend here that he's not with us still, he's still alive, who was also a key discoverer and researcher in the discovery of the gene for cystic fibrosis and I believe Huntington's disease. Being that you are in a Christian school, it's also an interesting read simply because Francis Collins is also a Christian, so you might want to check that out. Another one that I found absolutely fascinating, this just blows your mind, The Violinist's Thumb. If you're into chemistry at all, he also wrote one called The Disappearing Spoon. Sam Keen, some amazing stories about the discovery and the people involved in nucleic acids. So I'm just going to take the liberty of reading you just the first paragraph in the foreword here, just to give you a feel for some of the stuff that's going on in this book. Did the human race almost go extinct? Can genetics explain a cat lady's obsessive love for felines? How does DNA lead to people with no fingerprints and children born with tails? What can our genes tell us about John F. Kennedy's bronze skin? It wasn't just a tan. And Einstein's remarkable genius. And how did the right combination of genes create the exceptionally flexible thumbs and fingers of a truly singular violinist? And on it goes. So these books are all available in the school library. Don't be afraid to read. Good stuff. I would also, by the way, if you're motivated by such things, find some way of rewarding you for doing this little extra bonus type reading. There's one last picture on this screen, and it is what we might say is Chargaff's rules, or at least what it was Chargaff's rules were based on. If you look at the data where we've taken DNA and simply analyzed the proportions by percent of the bases adenine, thymine, guanine, and cytosine, if you look at that data, you can almost come to the same conclusion that Chargaff came to, and from that, the same conclusion that Watson and Crick came to in terms of how and which bases must fit together. And I think, take a look at it for a sec. In humans, the base percentage of adenine is about 31%, thymine 29%, guanine 20%, cytosine 20%. If you take into account experimental error, your conclusion should be that for all intensive purposes, the base percentages in DNA of A and T are equal. Same thing with G and C. One of the more likely explanations of why that is, of course, is that A and T are actually joined in DNA, G and C are joined. So there is actually a lot of experimental research that goes into this. So I'd encourage you to go through the DNA CD presentation that actually goes into some of the experimental background to this. I must confess my own interest in molecular genetics probably was given a kick in the backside when I pulled out of Vancouver Province a few years ago, and the cover of the Vancouver Province paper said, "Revealed the secrets of who we are." And what it was, of course, was the culmination of the Human Genome Project, but it had a little subtext to it. The most amazing story of human life, how we got here, where we're going, is revealed by the map of our genes. And got me thinking that a lot of the attributes we tend to think of in children, or in your case, future children, actually are genetically controlled. And by knowing the actual genes on the specific chromosomes, we now seem to have this window into very much our future as well as our children's future. So I got thinking hysterically absurd ideas about, hey, wouldn't it be nice to actually have a 7'4" daughter that could play a medal in volleyball, or have incredible skills on the piano or other intellectual skills, because we now know where certain genes are that code for these. Probably more realistic is just the realization that for our children and ourselves, we would like to avoid things like cancer and dementia and a whole lot of other nasty diseases. So yeah, the Human Genome Project is at least the first step in coming to a far better understanding of what those genes code for, and then ultimately perhaps having some control over them. So in a sense, I look at DNA as essentially a barcode. A barcode, you go through a check stand in your favorite supermarket, cashier goes beep, and the computer immediately knows that you've just bought a certain product and immediately orders a new one and replaces certain orders. Just a ton of information contained in the sequence of these bars. But if we think a barcode contains information, the information coded in your DNA molecule is just so much more significant. The point is that when that code goes wrong, we have a mutation, and mutations can lead to a whole lot of disorders. And the ones we tend to think of are the ones that turn up in circus sideshows, the bearded lady and that sort of thing. But the truth of the matter is, if you take a chromosome like chromosome 9, got a certain cancer at one location on that chromosome. By the way, the fact that these are shown as single chromatids tells you that these are chromosomes that haven't undergone replication yet. Prior to cell division, the cell is in a stage where it's simply carrying out its function and hasn't entered the synthesis stage where replication and protein synthesis is going to follow. So each chromosome now, following the Human Genome Project, has a map that shows us that certain types of diabetes, breast cancer, diseases like phenylketonuria, the inability to synthesize, not synthesize, but process the amino acid phenylalanine, a lot of diseases that are going to affect lifestyle are actually controlled by the sequence of genes, a code, right? So this has huge consequences socially, economically. If this information is actually recorded someplace, and you can basically find out your own genetic code by sending a small saliva sample to any number of labs around the world now. The question is if you really want to know that information. Once you know that you're likely to come down with Alzheimer's, but there's no cure for Alzheimer's, does that really improve the quality of your life? The big question again too is, where is God in all this, and are we really living in a deterministic society where we really have little control over our actions, our children's actions, who they are, who we are? I choose to think not, but the fact remains that if this information is contained in a repository, say in your schools, your universities, your future employers, is it likely that you as an employer are going to hire somebody that has potential, Huntington's chorea or cystic fibrosis, or you see long-term that they're likely to develop heart conditions, or all of these things are going to cost your company money. So what we have is deliberate choices by corporations to not hire certain people, and you can imagine the consequences in society of that sort of thing. But this is Biology 12, and we are not going to belabor the whole question of ethics here. You do have to know how protein synthesis takes place, but the importance of that protein synthesis really remains these bigger issues. So what you're going to get here is not sufficient to understanding the major processes of protein synthesis. In your study guide, you're going to see links to a number of videos that you really, really, really must check out. In a flipped classroom, you are going to get the actual full meal deal picture you need by watching crash courses, Bozeman Science, and other links that will actually show you animations, processes in actions, and so much better than just looking at pictures. I will go through just a brief outline of what this is all about, finish up with the protein synthesis package that you need to turn in, but again, I encourage you to you may think of going through those videos as a waste of your time. The fact of the matter is it's actually the best possible use of your time. So we're talking about the Human Genome Project. Obviously, we're talking about genes. So what the heck is a gene? The concept of a gene, everything that involves energy in your body, as you probably know by now, is controlled by a huge number of enzymes. We've done a chapter on enzymes already, and you know that there are thousands of them in each cell, that without them, reactions don't go from substrate to product. So it's the genes that actually code for the production of those enzymes. Many of these enzymes are, of course, proteins, and that's what DNA codes for, is protein. These genes can be regulated, and again, that's controlled, again, by genes, other genes. Other factors can regulate the activity of enzymes after production. The point is, the big picture here is that you start off with a DNA code, a double helix, represented by the sequence of the bases. DNA, of course, consists of rungs of a ladder that are held together by hydrogen bonds. These rungs of the ladder are basically nitrogenous bases. The nitrogenous base thymine, always paired with adenine, guanine, always paired with cytosine. Along the sides of the ladder, you have a sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. At some point or other, that sequence of bases on the DNA has to be read and transcribed into a different nucleic acid, messenger RNA. So mRNA stands for messenger RNA. Deoxyribonucleic acid has the master code. It is the recipe essentially for life and every protein and enzyme in your body. But at some point or other, when your cell needs it, a section of that DNA is going to be opened and read. You may be familiar with the term scribe. To scribe is to write. So what transcription is all about is taking that code and putting it in a form that can actually leave the nucleus. DNA is in the nucleus. mRNA leaves the nucleus and eventually attaches to a ribosome where the last stage of protein synthesis takes place, translation. The whole thing is about making protein, right? So our code that is transcribed now has to be translated into protein. And that's where the rubber hits the road. That's where the metabolism really comes in. So more will be said about this process, but for now realize that before this ever takes place, DNA makes a copy of itself. You're not going to mess with the master code. You want to have a backup copy. So the actual copy that is used for transcription, you undergo replication first. So the three processes: replication of the DNA, then transcription, finally translation. This is simply a fleshed out version of the previous diagram. Again, just emphasizing that it's genes that code for the production of proteins, which we mentioned are enzymes usually. So you start with the DNA code. At some point or other, that code has to be transcribed into messenger RNA so it can leave the nucleus and actually attach to a ribosome where the protein is actually made. So if you have a thymine nucleotide on the DNA, that means the complementary base is going to be on the messenger RNA. T always pairs with A, so mRNA is going to have an A there in that position. If you have a G in the position on the template strand on DNA, then you're going to have a cytosine, or C, on the messenger RNA. You may notice on the messenger RNA there is no thymine. So no T. And the way I remember that is the word not. So no thymine in messenger RNA. So everywhere you have an adenine on the DNA molecule, you're now going to have adenine pairing with another nitrogenous base called uracil. U stands for uracil. And you're not going to find uracil in DNA, but you are going to find it instead of thymine in messenger RNA. And again, that messenger RNA leaves the nucleus, moves to the cytoplasm where it attaches to a ribosome. There's all these amino acids attached to what are called tRNA, or transfer RNA molecules. They will attach to the three bases called codons on the messenger RNA and make a protein. And those proteins could be structural, they could be regulatory, contractile, things like antibodies, transport proteins like the ones in your cell membrane, or enzymes, catalytic proteins. So we have said that genes control protein synthesis. Where do you find the genes? Genes are found on the chromosomes in the nucleus. So again, chromosomes normally are not nearly as pretty as these sweet little artistic versions here. They're more likely like this. When they're not paired, they exist as single chromatids. The banding patterns really are where you're going to find the genes. We're not going to go into a whole lot of detail here because a lot of this you may already be familiar with, but DNA is usually not found on its own. Before cell division, it's found loosely as chromatin. Just before cell division starts, however, it condenses and it actually attaches to what are called histone proteins and forms these coils. And again, the purpose of that, it's believed, is to basically save space within a nucleus. A nucleus is not that big, and you have an entirely massive tome of information that has to be somehow stored within that. So all of the packaging and compression that can possibly go into it is applied at that point. So a replicated chromosome is double-stranded like this. The non-replicated chromosome simply exists as chromatin. When you look at a cell, even a human cell undergoing cell division, mitosis, you are going to see a whole lot of chromosomes that aren't nicely paired like these. So in order to look and study chromosomes, you have to do what's called karyotyping. Karyotypes display the chromosome contents of a cell organized according to their number, size, and type. So you eventually pair chromosome 1 pair of chromosome 2 based on their size, and you're going to notice that they have exact copies at the same locations of the gene. One comes from your mommy, one comes from your daddy. Normal human body cells, these are called somatic cells, have a karyotype with 46 chromosomes, existing as 23 pairs. Of those 23 pairs, 22 are called autosomes, and then depending on whether you are a female or a male, you are going to have one pair of sex chromosomes. So in the case of this guy is a guy, he's a male, he's got this little broken chromosome called a Y chromosome, and there's a whole lot of interesting information about what in the world genes are on that and why it looks so small and all the rest. But read, the violin book, and you'll find out a whole lot more about all of that interesting information. Just to quickly look at a human female karyotype though, every cell in a normal human cell except egg cells, and of course the reason for that is egg cells undergo meiosis, so you have half the chromosome number, you have 44 autosomes and 2 sex chromosomes. So these are the 2 X chromosomes in the female, and in the case of the male we have again 44 autosomes, 2 sex chromosomes, 1 X chromosome. This is of course why there's a lot of genetic diseases that are only shown up in males. Sex-linked diseases like color blindness, where it's passed from the mother to the male son, is going to is going to actually express itself because the male only has 1 X chromosome that he's got from his mother, right? The other one is a Y. So, back to DNA. DNA is made up of nucleotides. The building blocks of nucleic acids are nucleotides. And we're talking about nucleotides whether we're talking about DNA or RNA. All nucleotides have a sugar. In the case of DNA, it's deoxyribose. In the case of RNA, it's ribose. They all have a phosphate group, which of course is actually this PO4. And they all will have one of 4 different types of base. Adenine, cytosine, guanine, thymine. A closer look at a nucleotide. Again, you see the phosphate, the sugar, attached to the purine base guanine. It's not a big thing in this course to distinguish pyrimidine bases from the purine bases. The way I tell the difference is you'll notice that pyrimidines have the letter Y in it, cytosine, thymine, right? So just like pyrimidine, Y, Y, Y. And the purines, of course, like adenine and guanine, don't have a Y. You may wonder what these bonds are here connecting the nitrogenous bases. Those are hydrogen bonds. Those are relatively weak bonds because during replication, during transcription, they actually have to be unzipped and reattached, and they can reform rather easily. The genetic code is actually based on the sequence of amino acids. So the code is read in a sequence of 3 bases. They're simply called triplets on DNA. But when you have those triplets transcribed into messenger RNA, we call them codons. So you will get something like ACU or GCC. All of those are considered codons, and you will always be given a chart in order to translate those codons. And the only way you can translate the codons is by having amino acids that actually match these. And they, of course, are found on the transfer RNA molecule, which contains something called an anticodon. More to be said later. You see why you want to watch the animations. You see why you want to watch those Campbell activities. You need to see this in action. So each triplet codes for 1 amino acid, but more than 1 triplet may encode some amino acids. The code is said to be degenerate or redundant because you have more than 1 possible code, which is actually a good thing. If you look at your chart of codons, you'll notice that if you had a mutation where the last base was actually changed, often you'll still get the same amino acid. Then there are actually some codons which don't code for amino acids. They are codons that tell the cell to actually start, which is AUG, and then there are codons which actually stop the protein so you don't keep adding amino acids. So an amino acid added to an amino acid, joined to amino acid, of course, is a polypeptide. At some point or other, you have to stop the addition of the amino acids, or you're going to end up with the wrong protein. So below is a messenger RNA strand coding for 6 amino acids, 1, 2, 3, 4, 5, 6, and AUG would be the start codon. UAA, or methionine, would be the stop codon. Before you ever make a copy, before you ever actually start protein synthesis and begin to transcribe a sequence of bases on DNA, you want to make sure that you have a backup copy. So DNA always makes a copy of itself. It's called replication. This takes place in the nucleus. DNA is replicated to produce an exact copy of a chromosome in preparation for cell division. The first step requires that the coiled DNA is allowed to uncoil by creating a swivel point. This is a replication fork. The swivel point basically is created by enzymes, and there's a whole lot of enzymes that, quite honestly, you're not really terribly responsible for. I'll show you some of them here, but yeah, don't start hyperventilating, because the truth of the matter is you only need to understand that these steps in each of these processes is controlled by enzymes, but only very few of them would you be considered responsible for. So I'm not going to go into a whole lot of detail on this replication. You can look at the diagram here yourself. You can read as well as I can. It's much better to see this process of replication, the process of transcription and translation all in animation, rather than simply relying on a two-dimensional diagram. The point is, when replication ends, you now have 2 new strands of DNA which are identical. So everywhere you had a guanine, you're going to have a cytosine. Everywhere you had a cytosine, you're going to have a guanine. The reason this happens, of course, is you have a whole lot of free-floating nucleotides, one with thymine attached, one with cytosine attached, adenine, etc., floating in the nucleoplasm of the nucleus. If you get nothing else out of this section here, just make sure you know the base pairing rules, that adenine always pairs with thymine, guanine always pairs with cytosine. Now, did I mention there were enzymes involved in replication? These, of course, are a few of them: helicase, RNA polymerase, DNA polymerase 3, DNA polymerase 1, DNA ligase. Again, I wouldn't sweat the small stuff here. You will get a general sense of what these enzymes do and their importance, in particular on the lagging strand in replication, whereas the leading strand isn't nearly as complicated. There are 2 strands in DNA, right? So if you are going to synthesize new strands, you always have to do them in what's called the 5 to 3 prime direction. And this all comes from the fact that if you look at a DNA molecule, they're actually running anti-parallel. One end you have a 5 prime carbon, at the other end you have a 3 prime, and the only way that the bases can attach is if the other strand is running the other direction. So that may sound terribly confusing, but what it comes down to is the fact that the sugar in DNA, deoxyribose, is carbon 1, 2, 3, 4, 5. So on one side is carbon 5, that's the 5 prime end, where the next nucleotide is attached is the 3 prime end. So the way I remember it is, ask yourself, where are you more likely to be, or when are you more likely to be at school? At 3 to 5 or 5 to 3? And of course it's 5 to 3, so the direction of synthesis is always in the 5 prime to 3 prime direction. So these enzymes have no problem adding nucleotides in that direction. You actually get a continuous strand of newly formed DNA on that parental strand. On the other hand, helicase unzips the DNA strand, and you're going to need RNA polymerase to add a primer. Eventually you're going to need DNA ligase to join things. I'm going to give you a very, very brief overview of just some of these enzymes, but again, don't feel overwhelmed by what you see here. This is just for your general understanding. So, yeah. Helicase. This is the enzyme which splits and unwinds the two strands of DNA molecule. And again, notice they're running 3 to 5 and 5 to 3. So the actual new strand is going to form this direction in the 3' to 5' direction. RNA polymerase makes a short RNA primer, which is later removed. DNA polymerase 3 extends the primer with short lengths of complementary DNA. Like I said, you want to watch the animation of this in the Campbell CDs, because otherwise can be a little confusing. So again, DNA ligase is eventually going to join these fragments. These fragments, by the way, are called Okazaki fragments. On the lagging strand, you can't form it in the continual 3 to 5 direction. You only can do it in little chunks and then join those chunks. Okazaki fragments, I like that word. The term gene expression refers to the fact that these codes have to at some point or other be expressed in terms of protein. I think we've already covered this diagram, so I'm just going to move on. The next process, after you have a backup copy of DNA, a replicated copy, the next stage is transcription, and again you have enzymes involved in transcription. RNA polymerase is the big one there. The point here is that it's not the entire DNA molecule which is being read and transcribed. It's only going to be a section of it, a certain gene. So transcription is simply the formation of a messenger RNA strand formed using the DNA molecule as a template. And free nucleotides with bases complementary to the DNA are joined together by the enzyme RNA polymerase. So down here you're going to notice everywhere you have a C, you're going to have a guanine, the complementary base on mRNA. Everywhere you had an A, you're going to have a uracil or U on the mRNA. G with C with G, G with C, on and on it goes until you have the formation of a single strand of messenger RNA that is complementary to the template strand. So this original coding strand, and then you have the template strand. Of course, once that's completed, you're going to end up again with a recoiling of the actual molecule. So what happens after you have messenger RNA? Messenger RNA is a copy of the code. Code is for making protein. The protein is made not in the nucleus, but in the cytoplasm, in particular at the ribosome. So somehow that messenger RNA must leave through a nuclear pore and actually attach to ribosomes in the cytoplasm. The process of translation, and the word itself says what this is all about. It's translating the code that mRNA is back into an actual protein based on the original recipe. Translation is the process of building a protein or a polypeptide chain from amino acids guided by the codons, those sequence of 3 bases on the messenger RNA. So if you're ever asked to determine what the sequence of a protein is, you're looking at the messenger RNA, not the DNA, not the tRNA. So requirements for translation: you need messenger RNA because messenger RNA carries the code from the DNA that will be translated into the amino acid sequence. You also need for translation transfer RNA. Transfer RNA is responsible for transporting amino acids to their correct position on the mRNA strand. And of course, you actually need the factories as well that are going to take or undergo the translation, and these factories are ribosomes. Finally, in order to make this happen, you're making proteins, so proteins are always amino acids joined by peptide bonds, so you're going to have a supply of those amino acids in the cytoplasm, and that's why you eat. If you eat protein, you have amino acids available to make other proteins. So The ribosome itself is made up of a large subunit and a small subunit. No big deal in this course about any more of that. If you look at the actual molecule itself, you'll see it is a nucleic acid. You have an attachment site where an amino acid, so an amino acid would actually be attached here, and then you actually would have what's called the anticodon that is going to pair with the codon on the messenger RNA. You'll see how this is done in the protein synthesis package that you are supposed to do. So the anticodon is the site of the 3-base sequence that recognizes and matches up with a codon on the mRNA molecule. The actual sequence of events in translation, we tend to think of it as a 3-step process. Sometimes you'll hear the term initiation. Then you hear basically a term called elongation. Elongation is simply the polypeptide getting bigger and bigger as you add more amino acids. And then finally this process has to end because all proteins actually have a finite number of amino acids. Termination. Initiation, elongation, and termination. So, transfer RNA molecules with amino acids and the small ribosomal subunit attach to the codons on the messenger RNA strand. The large ribosomal subunit binds to complete the protein synthesizing complex, and then the ribosome moves along the mRNA strand. This process involves binding of the correct tRNA to a second site on the ribosome, formation of a peptide bond between the amino acids, and movement of the ribosome to the next codon. The final stage of protein synthesis, termination, occurs when the ribosome reaches a stop codon. This triggers the release of the polypeptide chain. The ribosomal unit falls apart so that they can be recycled and reassembled on a new mRNA strand. Cells are very, very efficient. So this would be an overview simply showing the ribosomes moving along the messenger RNA strand. Everywhere you see a 3 sequence of bases on the mRNA, call that a codon, and that codon is going to match the anticodon on the tRNA. So these are all tRNA molecules free-floating in the cytoplasm. Notice that they have at one end an amino acid actually attached. These are just abbreviations for lysine, serine, tyrosine, arginine, etc. This nasty looking picture is a summary of protein synthesis. This whole chapter, chapter 25, is about protein synthesis. You might want to, at some point or other, sit there, stare at this screen, and just kind of go through the steps, recognize the structures, and begin to hopefully feel comfortable with the general process. You'll notice that the enzymes have not been included in this diagram because it's already bloody messy as it is. So let's move on to the final reason you actually came here. And just before we do that, just a quick summary of what came before. We started with a DNA strand running 3' to 5'. On that DNA strand, you had bases, adenine, cytosine, cytosine. Notice that at some point or other, that sequence is going to be transcribed during the process of transcription into a complementary strand of messenger RNA. So the messenger RNA strand will run 5' to 3'. Everywhere you had an A, you'll have a U. Everywhere you have a C, you'll have a G. Everywhere you have a C, you'll have a G. That sequence of 3 bases on the mRNA is called a codon. That codon matches the tRNA's anticodon, and we have translation at the ribosome. So UGG stands for trip, TRP. UUU stands for phenylalanine, GGC stands for glycine, and on we go. Anyhow, you need some practice. That's the protein synthesis package. I'm just going to quickly zip through a few of these that may or may not cause you some pain. I think most of you could, at this point, muddle your way through this, but in order to save time and provide just a tad bit of clarity, I'll do a few of these for you, and then you can finish it off all on your own. So if you turn to the Dry Lab number 1 questions, they give you a base sequence of one strand of DNA molecule, AAT, etc. Give the base sequence of the complementary DNA strand. This would be the it's a double helix. This would be the other strand that is attached to it. And as you know, A pairs with T. So you're going to have T, T, A. And I like to do these in sequences of 3s just to keep my brain from getting too confused. GCC is going to basically be paired with CGG. AGT is going to be paired with thymine for adenine, cytosine for guanine, adenine for thymine. And then we get to the next one. GGT is going to be CCA. TGC is going to be AGC. And the last 3 bases, CAC, will give you on its complementary strand GTG. Then it says draw this DNA molecule. Guess what? They're not expecting some beautiful 3-dimensional diagram here. They're basically expecting you to realize that DNA is a double helix formed by a deoxyribose sugar joined to a phosphate, joined to a deoxyribose nucleic acid sugar, joined to another phosphate. And it's unfortunate that P's and D's look the same, but that phosphate is joined to another deoxyribose, which is joined to another phosphate, which is joined to another deoxyribose. You get the bloody point. That, of course, is just the sides of the DNA molecule. And, of course, in a DNA molecule, you have 2 sides. So guess what's down this side? Deoxyribose, phosphate. Deoxyribose, phosphate. Deoxyribose, phosphate. Deoxyribose, and by the way, on and on it goes. I don't think we really need to do the whole thing as long as you give me a few bases. The bases now make up the rungs of the ladder, and in this particular molecule it says draw this DNA molecule. A is on the one side, and what is it connected to? T, and then that T is connected to the D. And then the next deoxyribose is again A, and that would of course be attached to another T, losing my voice. And the third base is thymine. It is paired with, of course, adenine. And then the next one would be guanine. Oops. Connected to cytosine. And just in case you ever wondered, these are actually joined by what are called hydrogen bonds. So you might want to just throw a few little dots in between the bases, T and A, etc. That takes care of number 2. What about number 3? Give the base sequence of the strand of mRNA read from the original DNA strand. So this is the original DNA strand. The mRNA, everywhere you have an A, normally in DNA you'd have a T, but there's no T in messenger RNA. So the mRNA molecule is going to be U, A, and GCC will be CGG. AGT is going to be UCA. GGT is CCA. TCG is AGC. And then CAC would be GUG. Maybe just take a look back there, make sure you don't see any thymines, and you know you're on the right track. Number 4, draw this mRNA molecule. The difference between drawing DNA and RNA is that, of course, DNA is double-stranded, whereas RNA is single-stranded. And again, there's a different sugar. So instead of deoxyribonucleic acid, you have ribonucleic acid with the sugar ribose connected to a phosphate, connected to ribose, connected to a phosphate, connected to ribose, connected to a phosphate. This is so exciting. And then again, just like the DNA, you actually have to have attached to the sugars the correct nitrogenous base. So in this case, it would be uracil. And then it's uracil again. And then it's adenine. And then it's cytosine. And we'll forgive you if you don't really want to finish that, as long as you kind of recognize the major differences. Number 5, using the 3-base sequence codon, determine what amino acids this mRNA molecule codes for. You need to use one of these. This is called the genetic code. You cannot do these unless you have a chart like this in front of you. You are always given one of these on the test. So, using the 3-base sequence, determine what amino acids this mRNA molecule codes for. So if you're ever looking for the amino acids, you always have to look at the mRNA strand. You don't go back to the DNA, you look at the mRNA, because this is what's left the nucleus and actually attached to the ribosome. So, UUA. What is UUA? If I look for UUA, let me see now. UUA, there it is, leucine. You'll be pleased to know that these have abbreviations, but if you don't know them, you probably should actually write the word leucine. It's Leu, by the way, if you want the abbreviation. The leucine is attached to CGG. What is CGG? It may take us a while. CGG, where are you? Arginine, which we're going to abbreviate as ARG. And then the next sequence, UCA, yeah, serine, which is abbreviated SEER. And again, these are amino acids joined by peptide bonds. We have a polypeptide forming a small protein. Following serine, we have CCA. CCA is proline, which is abbreviated PRO. AGC would be AGC, yeah, serine again. Notice there's more than one codon for these things. And then finally, GUG is valine, which is val. Number 6, if the 4th nucleotide in the original DNA strand were changed from G to C, what sequence of mRNA would result? So, 4th nucleotide, 1, 2, 3, 4. That G is now been knocked out because of a mutation. This person has been living in their microwave oven for 12 years. We are now going to get a different protein. This is probably going to cause some big pimple on the guy's nose, whatever, because we have the wrong sequence of bases. The 4th nucleotide in the original DNA strand were changed from G to C. What sequence of mRNA strand would result? You basically go through this whole step again. And because I love you, you're going to get U, A, C, C, G, whoa, G, U, C, A, C, C, A, G, C, G, U, and then you're going to have a single amino acid at the end, G. And then what you do in number 7, what sequence of amino acids would this code for? You basically look at your chart again over here, and what you're going to do is UUA is leucine. Then the next one is going to be protein. You know what? I think it's time you actually did some of this yourself. I'm going to mention to you that there are certain questions here that you are not actually going to have to worry about. I'll give you a for instance. Yeah, I think you can do these without any problem. If you have a question about them, please, please, please ask in class, and I would be delighted to help you with it. Looking at dry lab number 2, very similar process. What is the base sequence of bases on the complementary DNA strand? Yeah, you do a whole lot of the same stuff that you did on the previous page. When you get to questions about actually drawing the amino acids, like this guy in number 4, guess what? Omit. In this course, you are not responsible for drawing actual long chains of structural diagrams in organic chemistry. I think you will be fine. So that would go for any other question like that. Number 4, when it says draw, omit. And of course, that would go for number 7 as well. Where are we? Omit. We're just about there, people. Take a deep breath. Yeah, there is finally a little quiz at the end just to see if you know what you're doing in terms of protein synthesis. Sometimes your chart is different than the one I was using earlier. You can use any chart you like it, because they basically all end up with the same amino acid. And then finally a summary page where you should be able to tell the difference between replication, where it takes place, what it produces, what this process is, again in the nucleus, what it produces, and then what this process is. And I'm going to be really kind to you. I'm going to tell you what this is. It takes place in the cytoplasm. And of course, what it produces is protein. It's an I, honest. This little guy got down here that you can't see is, of course, the tRNA molecule, tRNA. This would be the amino acid attachment. This would be the anticodon. So this is just a chain of amino acids. Amino acid chain, which we call a polypeptide. Ladies and gentlemen, you have survived. And the only thing else in this unit is, of course, some of the content in chapter 26. So I will mention that in BobbyTalks MolecularGenetics 2, which won't be nearly as painful and long as this one, but at some point or other you should check that out as well. We're done.